Tangwancharoen lab at Presention University, UCSD. Today I'm going to be talking about the genetic basis underlying thermal adaptation in the type 2 COVID part, divergence Californicus, focusing on the evolution of heat shock response gene regulation. So I'm generally interested in thermal adaptation. So temperature can affect biological and physiological processes, so that makes it a major driving force of the local adaptation. And different habitats, the temperature regime vary across different habitats that make the organisms that live in to become thermally adapted to their local environment. And my main big picture question is what are the mechanisms underlying thermal adaptation? And the system that I use is the type 2 fluid pot. Divergence Californicus is a good system to study heat stress response and thermal adaptation because they have a broad range from Baja California to southern Alaska, so that covers large gradient of the temperature gradient. And here's an example. We're trying to install a temperature locker in one of the typhoons here in San Diego, and these are the results showing that within a day, the temperature in typhoon can fluctuate more than 15 degrees Celsius, and sometimes they reach above 35 degrees Celsius. That's going to cause some acute heat stress. And previous studies have shown that different populations along the coast have different thermal tolerance phenotypes. So here, like populations from the south to the north, and you can see that the southern population survive at a higher temperature heat stress than the northern population. And in this study, I'm kind of focusing on the two populations. The heat tolerant southern population, San Diego, and the less tolerant Santa Cruz population. The following study looking at the transcriptomic response to heat stress in the two populations. Here's like the main results showing that in both populations, in response to heat stress, they upregulated groups of genes called heat shock protein genes. Heat shock proteins are chaperones that bind to the protein that's denatured by high temperature to help them pull back or prevent them from aggregate and cause damage in the cell. And the difference between the more tolerant population and the less tolerant one is the main is that even though they both upregulate heat shock protein, but the southern population upregulate at a higher magnitude than the northern population. So that's going to lead to my question for the study is that what is the genetic basis that underlying the pattern between the two populations. For the gene expression, there can be two main underlying factors. So that there are cis regulatory element and trans regulatory element. So cis regulatory element are the DNA sequence like the promoter or the enhancer, and the trans-regulatory element are the transcription factor that interact with cis elements in governing the gene expression. And my project is trying to tease out the evolution in cis and trans effects. And we can do that in the F1 hybrid. So the difference in gene, the allele-specific expression in hybrid only going to come from the cis effect because in F1 hybrid, you have both allele from the parents, and you have the two trans acting alleles acting on the same site. So the difference only going to come from the cis effect. So the way I design my experiment, so I use the two populations, San Diego and Santa Cruz, make a reciprocal hybrid cross, and I use the 50 adult individuals and put them under two treatments, so the control constant at 20 degrees Celsius and 1 hour heat stress at 35 degrees Celsius, and have like two replicates, and then extract DNA and sequencing with the Illumina HiC 4000 with single N 100 base pair. And once I got my reads back, I mapped it to the two population transcriptome reference previously published by Barreto et al. 2015 using the program CLC Genomic Workbench. And I filter out the genes with the low coverage and also the genes that can be differentiated between the two populations. So I end up I end up with the final number is like 9,853 genes that I use for further out analysis. So first part, for the allele-specific expression analysis, I use the unique reads, so that's the reads that only map to the one allele. And for that, I have to do a statistical test on the tree test. So first, so looking at the allelic imbalance in hybrids, that's gonna infer the cis effect. And the next one is just looking at the difference between the parent population. And the third test is to compare if the allele-specific expression ratio in parents and hybrids are the same or not. And I also use the total read to analyze for differential expression between control at 20 degrees C and heat stress 35 degrees C. The total reads are the reads, include a unique reads and the reads that can be differentiated between the two alleles. And so I'm going to show the result from one of the reciprocal cross. If you wonder about the other one, you can come find me later. So with the hybrid allele-specific expression, the main thing that I found that between the two treatments, the heat stress treatment has more bias expression than the control treatment. And when I include the hybrid expression and the parental expression, I can tease out the effect of the cis and trans. So here the hypothetical result that has been using by the study McManus et al. 2010. So if the genes don't have divergence in parents and in hybrid, they are conserved, like in the black area. If they show divergence in parent but not hybrid, that means it's only controlled by the trans effect, because in the F1 allele having all the same trans. And for the cis effect, so you see the different allele-specific expression in hybrid, and if the ratio is the same as in parent, so that's indicating the cis effect only. And the rest in green and yellow are the combination of both cis and trans. The yellow part means the compensatory, so in parents they don't show the divergence because the cis and trans effects have the opposite sign and interact with each other to conserve the expression pattern. And here's one of my results from one of the reciprocal cross and the heat stress. And most of the genes are conserved, but the genes that do show the allelic imbalance, a lot of them are underlined by the cis effect only. And so when I also looking at with the differential expression between the heat stress and control. So this is the log expression of the control and heat stress. So the dots above are upregulated, and the different color indicates the direction of the bias. And you can see here that there are more bias in the genes that are upregulated. And now I'm focusing on the group of genes I'm more interested, that is the heat shock protein genes. So they are all upregulated. And I'm going to focusing one in particular called heat shock protein beta 1. So in this gene, so it's upregulated after heat stress, and it shows like log ratio of the expression in hybrid is like about 1.5. That means like in hybrid they express the San Diego allele about like close to 3 times more than the Santa Cruz allele. That's a strong bias. And the test show the cis effect only. And also this gene only show the bias expression under the heat stress conditions. And all these patterns hold true for both the reciprocal cross. And besides that, in this gene in our lab, we have showing that it contributes to the survivorship after heat stress. So the recent study by Barreto et al. Has knocking down the expression of heat shock protein beta 1 using RNAi, and showing that with the knockdown animal, they increase of the mortality after heat stress. So it's an important gene. So for heat shock proteins, the heat shock protein gene regulation has been well studied since like the 80s and 90s. So there's heat shock transcription factor, or HSF, that got activated after heat stress and rise, and they bind to a specific motif called heat shock element, which is an inward repeat of the 5 base pair units that have a 3 base pair in the middle that's conserved, and the system is conserved from yeast throughout the Drosophila environment, so it's very highly conserved. And like the heat shock factor binds to the heat shock element and upregulate the heat shock protein genes. And back to the heat shock protein method one that I talked about earlier. So I looked up in the promoter region of the gene, and I found 2 clusters of the heat shock element here at this location. And in more heat tolerant southern population, you find what I call like the perfect heat shock element, that is like all the sequence according to the same as the consensus of the heat shock element. And in the less tolerant northern population, there's a substitution in the conserved motif of the heat shock element. And so that I kind of hypothesized that it might affect to the binding efficiency of the heat shock transcription factor that leads to the difference in gene expression, and consequently lead to difference in thermal tolerance between 2 populations. So in summary, divergence in gene expression can contribute to local adaptation, and the allele-specific experiment can tease out the effect of resistant trans, and we hypothesize that it's involved with the heat shock factor regulatory network, and the mutation in the heat shock element might affect the divergence. And I'd like to thank people from my lab, especially Tiago and Tessa, who helped me a lot with the informatics, and I'll take any questions. Yeah.
So are there any differences in the...
Yeah, there are some genes that only one, like only bias in one cross, and there's some like previous study that show that kind of can be like maternal effect, and maybe like mitochondria genotype that affect that too. Right?
Yeah. Did any other genes pop out as dysregulated?
Which genes?
Did other genes show? What other genes showed up conspicuously dysregulated?
I've looked at some. I've seen some of the like ubiquitin related gene and some. I couldn't think of. I can show you more about that.
Sure.
Sorry,
No problem.
I can't think of my head right now.
Sure, no problem.
The heat shock transcription factor, does that have any interesting evolutionary effects?
It's really highly conserved. I know that
differentially expressed.
Oh, differentially expressed. In this data set, no, and it's not biased also. So it's, yeah.
Can you use gypsy?
We're trying to do that next. Actually, yes, it will be good to have the sheet to control like the binding data, right? That will prove that the hypothesis is true.